Hey GCSE biologists, now in this video we're covering all of the extra triple science content for Module 5 GCSE biology, and there's absolutely loads of it, and this is probably some of the hardest stuff you're going to be learning about in GCSE, so make sure you watch the full video and take notes. Also make sure you click the link in the description so you can access more free flashcards that we will send to you immediately, covering all of this triple science content that I will cover in the video. Okay, let's get started. In this video, we're looking at Module 5 again, but this is all of the triple science content for Module 5. There's loads of it. We're going to be looking at the brain, the eye, the kidney, temperature regulation and tropisms, all of that good stuff. This is for those of you that are doing separate sciences, so make sure you watch this video. This is some really hard GCSE content. The first thing we have to look at is the brain, and you do have to be able to label the main structures in the brain. So the outer portion is known as the cerebrum, or you can call that the cerebral cortex. So that is this whole kind of outer portion of the brain here. Then we've got the hypothalamus, which is in the middle of the brain. Next to the hypothalamus, you've got the pituitary gland, which is a small P-shaped gland, which is also in the middle of the brain, kind of right next to the hypothalamus. At the back, this portion here is the cerebellum. Don't confuse that with the cerebral cortex or the cerebrum. They sound very similar, but that is the cerebellum at the back of the brain here. And then just at the top of the brain stem, kind of here, you've got the medulla, or the medulla oblongata, and then obviously running down your spine, you've got your spinal cord, and the brain and the spinal cord together make up your central nervous system. Now, once you know how to label it, you need to know the functions of these key parts of the brain. Just really simply, but the outer portion of the brain that we call the cerebrum, that controls your conscious thoughts. So it's involved in memory, remembering things, intelligence, learning a language, things that you have to think about, learn. So when you're studying, when you're watching this video, you are using your cerebrum. The cerebellum, which was the back part of the brain that we spoke about here, coordinates muscular activity and balance. So the example I always use is when you're riding a bike, because you're using muscle activity and you're having to balance. The medulla, or the medulla oblongata, is involved in unconscious activities, so things that you don't have to think about. This is actually part of your autonomic nervous system. So things like control of your heart rate or your heartbeat and your breathing rate that you don't consciously think about. That will be controlled by your medulla. The hypothalamus is involved in homeostasis. You should know what that is. So maintaining a stable internal environment to provide the optimum conditions for enzymes and cell function. So for example, it's involved in temperature regulation. It's also involved in maintaining the concentration of water in your blood. So it's linked to homeostatic mechanisms. And then the pituitary gland, which we covered when we did the endocrine system in the first part of this video, is known as the master gland. Its job is to release hormones, but a lot of these hormones control other glands, which is why we call it the master gland. So it releases hormones such as FSH and LH, which are hormones involved in the female menstrual cycle. It releases TSH, or thyroid stimulating hormone, that stimulates the thyroid gland to release thyroxine. It also releases ADH, or antidiuretic hormone, that we're going to be learning about later in this video. That hormone controls the water reabsorption from your kidney tubules, so it will affect the volume and the concentration of urine that you produce. Now, if you're higher tier and you're taking triple science, which I know most of you will be, you need to look at ways that we can study the brain. Now, we call them neuroscientists, scientists who study the brain. And we need to know 3 examples of how they can study the brain and learn more about the functions of the different parts. So the first way they can do that is to study people with brain damage, because obviously if part of the brain has been damaged, they can look at the effects of that on the person to figure out what that part of the brain must have been involved in. We can also use electrical stimulation, or you can talk about using electrodes. Electrodes can be placed onto the scalp or directly onto the brain, and we can stimulate different parts of the brain to see what the response is and therefore what that part of the brain is responsible for. We can also use the electrodes to monitor electrical activity, and this does help us to diagnose conditions such as epilepsy. The third method of studying the brain is to use an MRI scan. MRI scans can monitor the electrical activity in the brain. They provide us with detailed images of the brain structure, the brain activity. So they can use it to see which part of the patient's brain are active when they do different things. So if they listen to music, if they recall a memory, if they look at images which provoke emotion, which part of the brain is being stimulated, because therefore it's telling us that that part of the brain is involved in that response. MRI scans can also be used to look for abnormalities in the brain, such as swellings, tumours, any blood clots or blockages, or any internal bleeding, because they do give us a detailed image of the brain and its structure. Now, in terms of what you need to know here, you definitely need to know the 3 examples of how we study the brain. But when these questions have come up in the past, you're usually given a little bit of information, like a bit of information about the MRI scan. So I suggest you just read through and have a little idea of what each of these types of study can be used for. Now, obviously, anytime we study the brain, there are potential risks. The brain is a very complex organ with billions of interconnected neurons. It's very hard to figure out what all of the different parts do. It's very hard to study the brain. And any question you get about brain surgery, obviously that has many risks. Potentially damaging the brain tissue, that can lead to a lifelong impact on a person's personality, memory, intelligence. So it's a really, really complex field of study. Now, the next part of the triple science spec for Module 5 is all about the eye. Again, you have to be able to label the main structures in the eye. So let's look at that first. At the back of the eye, we have the optic nerve that will send electrical impulses to the brain. And then the back surface of the eye, the back inner surface of the eye, is known as the retina. Towards the front of the eye, you've got the lens, which is this kind of oval shaped structure here. That can change shape. And the lens is connected to suspensory ligaments. So the suspensory ligaments hold the lens in place, and the suspensory ligaments will then connect to ciliary muscles. And the ciliary muscles can contract and relax to change the shape of the lens. At the very front of the eye, you've got the cornea, which will bend or refract the light. You've got the iris, which is the coloured part of your eye. And that can control the size of the pupil. The pupil is literally just the hole that lets light enter your eye, the hole in the middle of the iris. Now, we need to go through the parts in a little bit more detail than that. So once we can label them, we need to know the function of each part. So the retina is really important that you look at, because in the retina, which remember is the back surface of the eye, there are receptors or receptor cells for light. We call them photoreceptors. There's actually 2 types. They're called rod cells and cone cells, which you'll learn more about if you do A-level biology. But they detect any change in light intensity. Remember, a receptor is a cell that detects a stimulus. These receptor cells or photoreceptor cells detect a change in light intensity, and they convert that light into an electrical impulse, which will then be sent down the optic nerve at the back of the eye. The cornea, which is at the front of the eye, is transparent, so it lets light through, and it bends or it refracts the light as it enters the eye. The iris, which we said is the coloured part of your eye, contains muscles that help to control the diameter of the pupil. And we're going to look at this in much more detail because that is something you need to understand. The pupil is just the hole in the centre of the iris, and that's where light enters your eye. The lens can change shape. And the reason for that is so we can focus the light onto the retina so we can see images. So the lens can change shape, it can get fatter or more curved, thinner or less curved, to make sure that the light does get focused on the back of the eye or on the retina. Now, the lens is connected to the suspensory ligaments, and the suspensory ligaments are connected to the ciliary muscles. So these next two kind of work together. The ciliary muscles can contract or relax to change the shape of the lens, and the suspensory ligaments hold the lens in place. They work with the ciliary muscles during accommodation, and accommodation is a topic that we have to know in more detail. So again, we're going to look at that in this video. The optic nerve at the back of the eye transmits or carries electrical impulses from the retina, where we find the photoreceptors, to the brain. And the sclera, which is at the very, very front of the eye, it's not actually on my diagram, but the very kind of front layer of the eye, the sclera, is a tough outer layer that protects your eye. So again, that's really important. Okay, let's get into what we have to learn then about the eye. So we have to learn about the pupil reflex, which is basically how our eye adjusts to bright light or a higher light intensity, or dim light or a lower light intensity. So the first one we're going to look at is how it adjusts to bright light. Now, both of these responses involve muscles in your iris, the coloured part of your eye. And there are two sets of muscles in your iris. You have the radial muscles and the circular muscles. And that does make sense, because if you look at this iris over here, this nice green iris, the radial muscles are the ones that radiate out like rays of the sun. They're called the radial muscles. And the circular muscles are the ones that circle, circle the iris. So you've got your radial muscles and your circular muscles in the iris. Now, when you go into bright light, so maybe you walk into a room with really bright light or a high light intensity, the radial muscles in the iris will relax and the circular muscles in the iris will contract. Now, I actually think that's quite easy to remember because the letters match. So radial, relax, R and R. Circular, contract, C and C. And I know it's a bit silly, but I kind of think of that as like, well, this is in bright light, so the letters can see each other, so they match up as they should. So in bright light, R, R go together, C, C go together. So bright light, the radial muscles relax, the circular muscles contract. And if you can picture the circular muscles contracting like this, the pupil is going to constrict or get smaller, so less light enters, okay? So the pupil becomes smaller, less light will enter, and that will prevent any damage to your retina, because if too much light enters the eye, that can damage the retina. Now, once you've learned that, in dim light, it's just the opposite. So in dim light, the radial muscles contract and the circular muscles in the iris relax. And again, it's a little bit silly, but I think of it like it's dim light, it's quite dark, the letters can't see each other, so they don't match up. They don't go together. So we've got R and C and R, right? So the radial muscles contract, the circular muscles relax. And if you picture the circular muscles relaxing like this, the pupil will dilate or get bigger to let more light into the eye, as you can see here, to help you see in dim light. Okay, now key things to remember here, we're talking about the muscles in the iris. It's the muscles in the iris that control the size of your pupil. So bright light, radial muscles relax, circular muscles contract, pupil gets smaller, less light enters the eye, so we don't damage the retina. And then in dim light, it's the opposite. The letters do not match because it's dark, they can't see each other. So radial muscles contract, circular muscles relax, pupil dilates, or you can say it gets wider, it gets bigger to let more light in so we can see. So that's the first kind of tricky response when it comes to learning about the eye. That is the pupil reflex, and that is about adjusting to the light intensity. Now, the second thing, which is kind of similar, so pupils get it mixed up, it's a second response. This is called accommodation. This is not to do with light intensity. This is about focusing on near or distant objects. So focusing on something like your phone or a book that is very close versus focusing on something very far away, like you're looking into the distance at a tree or some scenery, or maybe your telly is really far away. So this is called accommodation. It's focusing on near versus distant objects. Now, this does not involve the iris, but it does involve some other muscles. So let's talk about it. The eye can change the shape of the lens to focus on near or distant objects. This is called accommodation. So it's the shape of the lens, which is that kind of oval shaped structure at the front of the eye. It's the shape of the lens that we're changing. And the lens is connected to suspensory ligaments, and suspensory ligaments connect to ciliary muscles. So here's my suspensory ligaments, and here are my ciliary muscles. So it's these three structures working together that are involved in the process of accommodation. The lens, which will change shape, and it will change shape because of the action of the ciliary muscles and the suspensory ligaments. These muscles are not the same as the circular muscles in the iris, okay? These are the muscles that connect to the suspensory ligaments and the lens. So let's go through how we would focus on a far away or a distant object. So the ciliary muscles would relax and the suspensory ligaments would tighten, and that basically pulls on the lens because the suspensory ligaments get tight and they pull on the lens, so the lens becomes less curved or thinner. This means with a thinner lens or a less curved lens, the light is refracted or bent by a lesser amount, so it focuses on the retina so you can see that distant object clearly. Now let's have a look at this on a diagram, okay, so you can see all the different parts. So the ciliary muscles will relax, that will pull the suspensory ligaments tighter. The lens then gets pulled so it becomes thinner or less curved, and if your lens is less curved, it bends the light less or refracts the light less, which helps it to focus at what we call the focal point on the retina. Now, once you've learnt one side of this story, you've learnt the other really, because you just say the opposite. So let's do the focusing on near objects. And this is the one that I actually remember off by heart. If you're focusing on something near, like your phone, and it's here, the ciliary muscles contract and the suspensory ligaments slacken. Now, I find this side of the story easier to remember. And again, it's super silly, but I'll share it with you because it might help you remember all of this. This is about near objects or close objects. So again, I think of the letters matching. If the objects are close or near, then the C is next to a C. The S is next to an S. So ciliary muscles contract. C goes with C, they're close together because the object you're looking at is close. Suspensory ligaments slacken, so the S goes with an S. They're close together because the object you're looking at is close. I've no idea if that's going to help you, but that's how I remember it. So close objects, C goes with C, ciliary muscles contract. S goes with S, suspensory ligaments slacken. Now, because the suspensory ligaments slacken or loosen, the lens becomes more curved or it becomes fatter. That will refract or bend the light more so it focuses on the retina. So let's have a look at this one. So this is close focus. The contracted ciliary muscles contract, suspensory ligaments slacken or loosen, so the lens is not pulled as much. So the lens stays rounder, or you can say more curved or fatter. And with a fatter lens, it is bending the light more, which again helps it to focus onto the retina where we want it to focus at that focal point so we can see clearly when the image is close up. So my suggestions to you would be to learn close focus, because that's where the letters match, close focus. And then you can just think, oh, if it's about distant focus, it's the opposite. Everything is the opposite. But I've written this like how a mark scheme would have it written. So you need to be using these kind of words. Basically, you need to be saying what happens to the ciliary muscles, what happens to the suspensory ligaments, what happens to the lens, and then how much is the light bent by so that it can focus on the retina. So that's accommodation. That's all about focusing on near versus distant objects. And remember, that involves the lens, the suspensory ligaments, and the ciliary muscles. Whereas the pupil reflex involves the muscles in the iris, the radial and the circular muscles. Let's talk about vision defects next. So problems with the eye. So we need to know about myopia. Now, myopia is short-sightedness. And again, I have so many silly ways to remember this, but myopia is a shorter word than hyperopia. So I remember myopia is a shorter word, it's short-sightedness. Now, if you're short-sighted, you can see near objects clearly, but distant objects are blurry. So you've got good short sight, you're short-sighted. You can see these objects, but you can't see things in the distance. This is because light will focus in front of your retina. And there can be a couple of reasons for this. It might be your eyeball, the shape of your eyeball is too long, or maybe your lens is too curved or too fat. So the light is being refracted or bent too much. Let me show you this on a diagram. So here, this person has normal vision, okay? So their eyeball is the right length, their lens is the right shape, so you can see that the light is being focused as it should be on the retina. Here is someone with myopia, short word, short-sightedness. So when they're looking at things in the distance, you can see that the light is focused in front of the retina. It is not hitting the retina or focusing on the retina as it should be. Now, this might be because their eyeball is too long, or maybe their lens is more curved or fatter. It's bending the light too much, so it is focusing in front of the retina. Now, we want the image to focus, or the light to focus on the retina, so you could correct this with concave lenses, which will spread out the light rays so they're not bent as much. Now, a concave lens is this shape. The lens is kind of sunken in on both sides, and that will be placed, obviously if they're wearing spectacles or glasses, it will be placed in front of the eye, so the light rays are not bent as much, so they do focus on the retina and they are able to see even at long distances. Now then we have hyperopia. Now I think hyperopia is a longer word, so this is long-sightedness. If you're long-sighted, you can see distant objects clearly, but near objects are blurry. So they're long-sighted, good long sight, can't see close up. This is because the light focuses behind the retina. So this might be because their eyeball is too short, which is why the light is focusing behind the retina, or maybe their lens is too thin, so the light is not being refracted or bent enough. And again, I'll show you it on a diagram. So normal vision, you can see the eyeball is the right shape, the lens is the right shape, and the light is focusing nicely on the retina, so you can see. But with hyperopia, their lens, it doesn't really show it on here, but I'll draw it for you. Their lens might be thinner. Maybe even their cornea is a little bit flatter as well, so it's not bending the light as much. Their eyeball might be shorter. If the eyeball is shorter or the lens is thinner and doesn't bend the light as much, then the light can be focused behind the retina. And again, if they're looking at close-up objects, they will not be able to see them clearly. This would be corrected with a convex lens to bend or refract the light more. So a convex lens curves out on both sides. So if they're wearing glasses, it would be here. It would bend the light more So you've got a sharper or a steeper bend, so it focuses onto the retina. Now, we're going to look next at controlling body temperature. This is an example of homeostasis, maintaining a stable internal environment, and obviously this is to provide optimum temperatures for all of our enzymes. Now, the optimum temperature for our enzymes is 37 degrees C. This is our human body temperature. And this is the temperature that enzymes will work best at, will have the fastest rate of reactions, will have the right number of collisions between enzymes and substrates and lots of enzyme-substrate complexes, so reactions in the body will happen quickly. So we want to keep it at around that temperature to provide the optimum temperature for our enzymes. If the body temperature got higher, enzymes may start to denature. They lose their shape, the shape of their active site changes. But if the body temperature gets too low, enzymes won't denature, but they will work more slowly because they'll have less kinetic energy and there'll be fewer collisions between enzymes and substrate. So that would reduce the rate of important metabolic reactions like respiration, for example. So how do we control our body temperature? It's actually a process known as thermoregulation, but you can call it temperature regulation. First of all, we need receptors, right? We always need receptors to detect a stimulus. Now, in this case, the stimulus is going to be a change in temperature. That's the change in environment, right? So whether that's an increase in temperature or a decrease in temperature, that will be monitored and detected by receptors. But what makes this topic a little bit complicated is we have these temperature receptors in two different places. These temperature receptors are known as thermoreceptors. You can call them temperature receptors, but they're located in two places. So we find them in our brain in what we call the thermoregulatory center. This is actually in our hypothalamus, which we labeled on the diagram of the brain earlier. So in the hypothalamus, in your brain, is the thermoregulatory center. That contains temperature receptors, but also your skin, all over your skin, all over your body, there are more temperature receptors. And the reason we need both of these is they monitor different things. So the thermoreceptors in the thermoregulatory center, which are located in the hypothalamus in the brain, they will monitor and detect any changes in the temperature of the blood that flows through the brain. We call that our core temperature, our inner temperature. So as blood is flowing through your brain, through your hypothalamus, the temperature receptors are monitoring the temperature of the blood, or the core temperature, and detecting any change in the temperature of that blood. Now, the thermoreceptors in the skin, they monitor the temperature of your skin and the external air temperature, and they will send electrical impulses to the thermoregulatory center. So the thermoregulatory center in the hypothalamus in the brain is the main coordinator because it's receiving information from its own receptors about the temperature of the blood flowing through the brain, but it's also receiving electrical impulses from the temperature receptors in the skin about the external temperature and your skin's temperature. So once it has all of that information about both the core temperature and the skin temperature, it's coordinating your body's response. So does the body need to warm up or does it need to cool down? So let's go through the stories. Now, the thermoregulatory center uses negative feedback. Remember, that means if the temperature increases, it will coordinate responses to lower it. If the temperature lowers, it will coordinate responses to raise it, to bring it back to normal temperature, which for us is body temperature at 37 degrees C. This ensures that the temperature remains stable, so it ensures homeostasis, a stable internal environment to provide optimum conditions for enzymes and cell functions. So let's imagine we've got normal body temperature to begin with, but then And that's what we want, right? We want normal body temperature, 37 degrees C. But then there's an increase in body temperature. So it's a hot day. If the body temperature increases, thermoreceptors in the hypothalamus and the thermoreceptors in our skin will detect this change. And remember, the thermoreceptors in the skin will send electrical impulses to the hypothalamus. So the hypothalamus has information on the skin temperature as well as the core or the blood temperature. If they detect that the body temperature has increased, there are two main things that will happen. Sweating, the production of sweat will start to increase, and vasodilation will start to take place. Now, vasodilation is basically a widening of arterioles, so narrow arteries. That will lead to a decrease in body temperature. So we're going back to normal body temperature, which is what we want. Okay, now the converse of this, if there is a decrease in body temperature, so it's a very, very cold day, we're outside, we don't have a coat on, the thermoreceptors in the hypothalamus, in the brain, and the thermoreceptors in the skin will detect this change. And remember, the thermoreceptors in the skin will send the electrical impulses into the thermoregulatory center in the hypothalamus, so it has all the information to coordinate the response. The two main things that will happen: shivering, which is basically really rapid muscle contraction, and vasoconstriction, which is a narrowing of arterioles. And that will lead to an increase in our body temperature to return our body temperature to normal. So you can see this is negative feedback. If there's any change away from the normal level, it is detected by receptors. Responses are coordinated to reverse that change. Now, what we need to do next is look at these responses in a lot more detail, because we haven't done enough yet for triple science. So if your body is too hot, so we've detected an increase in temperature, sweating will start to happen. So what do we need to know about that? Well, sweat glands in your skin will produce sweat. Sweat evaporates. And when sweat evaporates, it's transferring heat energy away from the skin. So it basically cools the blood, which cools the body. Because sweat, which is mainly water, it does require a lot of heat energy to get it to evaporate. And that heat energy will come from your body. It will come from the heat energy in your blood. So as it evaporates, it takes that heat energy from your blood, which will cool your blood, which will cool your body. Now, the second thing that happens if your body is too hot is vasodilation. Now, this is where blood vessels, it's actually the arterioles, so the very small kind of arteries, they will dilate. But we have to be a little bit careful here. It's not every blood vessel that's going to get wider. It's not every arteriole that's going to get wider. We have to say it's the blood vessels near to the skin surface that dilate or widen. Not the rest, just the ones near to your skin surface. Why? Well, if the blood vessels supplying your skin widen, that will increase blood flow to the skin surface. And if we increase blood flow to the skin surface, more heat energy will be lost through the skin, or more heat energy will radiate out through your skin. Let me show you this on a diagram. So we've got here an artery. Now, an artery branches into a smaller arteriole, which is kind of just like a mini artery. And these mini arteries, if you like, will dilate or become wider. That's what that means. The lumen widens. So more blood, if they get wider, more blood can flow close to your skin surface. And if more blood flows close to your skin surface, more heat energy can be lost from your blood through your skin surface. So more heat energy is lost through the skin surface. But you do have to be really careful to say it's blood vessels or arterioles near to the skin surface that dilate to increase blood flow to the skin surface, so more heat is lost through the skin. Now, obviously, if you're too cold, sweating will stop because we don't want any evaporation of sweat. So that's kind of a given. Sweating will stop. But other things that can happen, we can start to shiver. This is basically rapid muscle contraction. Now, when muscles contract really rapidly, sometimes even in your face, and you can feel your teeth like chattering, they increase their rate of respiration, which releases more heat energy to warm us up. Because contraction, rapid contraction, does require a lot of energy. So they have to do more respiration, which will release more heat energy, which warms us up. Now, then we have the opposite of vasodilation. We have vasoconstriction. So let's go through it again. Again, we say it's blood vessels or arterioles near the skin surface. They constrict or they narrow. The lumen gets narrower. This will reduce blood flow to the surface of the skin and reduce heat loss through the skin, or reduce heat loss by radiation. I'll show you a picture of vasoconstriction. So again, we've got the artery supplying blood to the skin. That branches into a smaller arteriole. It's actually the arterioles that constrict. So they've got a little bit narrower here. If they narrow, less blood flows close to the skin surface, so you can see there's less heat energy. In fact, it looks like there's no heat energy being lost here. There might still be some heat energy lost, but there'll be less heat energy lost through the skin surface. Okay, but again, make sure you're clear. It's the blood vessels near to the skin surface that constrict or narrow, which reduces blood flow to the skin surface, so less heat is lost through the skin. You have to also be careful not to say capillaries dilate or capillaries constrict. It's not the capillaries doing this. Think back to your paper 1 knowledge. Capillaries do not have muscle in their wall. They cannot constrict or dilate. It is not the capillaries. So either specify that it's arterioles, or if you think you're going to forget that, just say blood vessels near the skin surface. But do not say capillaries, because it is not the capillaries. Their walls are just a single layer of cells with no muscle. They cannot narrow or widen. Okay, let's have a look at probably the hardest bit in Module 5 for Triple Science. This is all about maintaining water and nitrogen balance. So it's going to take us into the kidney. Obviously, our body must maintain a constant water level for cells to function. Now, you should know from paper 1 that water moves in and out of cells by osmosis. Now, if the concentration of water in the blood changes, this would affect the movement of water in and out of cells by osmosis. So if you had too much water in your blood, so your blood was too dilute, water would enter your cells by osmosis and your cells may swell and burst because they're animal cells, they don't have cell walls. So if too much water were to enter your cells, they would swell and burst. But equally, if you had too little water in your blood, so your blood was more concentrated, water would leave your cells by osmosis and the cells would shrink and shrivel. So we do have to make sure we have the correct concentration of water in our blood. Let's think about all the different ways that we lose water from the body. So we lose water from our lungs when we exhale or breathe out, because water vapor is lost in exhale breath. Now, we can't control how much water vapor is lost when we breathe out. Water vapor is also lost by our skin when we sweat to try and cool ourselves down if we're too hot, because water evaporates from the skin surface, as we've just said, to take heat energy from your blood and help to cool the body. Again, we cannot control how much water we lose in our sweat. But the third way that water is lost from the body is through our kidneys, because they produce urine, and water is lost in our urine. And this can be controlled. We can control how much water we lose in our urine. It's controlled by the kidneys, and it's controlled by a hormone called ADH, which we're going to look at very shortly. Okay, so let's get into the kidneys. So the kidneys, you can see them here. You've got two kidneys, right? Kind of low down towards the back of your body. The kidney's job is to filter your blood and produce urine. Now, the blood flows into the kidneys and they filter the blood to remove certain substances from your bloodstream. They will remove all urea. Urea is a toxic waste product that does need to be excreted in the urine. They will remove mineral ions, such as sodium ions. They will remove water. They will remove glucose. Also, amino acids as well, because they're all small enough, these things, to pass through the filtering system in the kidneys and enter what we call the filtrate. So all of these things will be removed from your blood. These substances are small enough to pass through the filters in the kidneys. They will leave the blood and pass into the kidney tubules, forming the filtrate. Now, other things that are in your blood, such as proteins and your blood cells, like your red blood cells, are too large to pass through the filters, so they will remain in the blood. So let's have a look at this on a diagram. So here is your blood, or your blood plasma, the liquid part of your blood. This represents the filters in your kidney tubules. That's what we call them. They're called nephrons, but at GCSE you can just call them kidney tubules. So they have like these partially permeable membranes that act as filters. Now, anything small enough is going to get filtered out of the blood in your kidneys and will form what we call the filtrate, which is in the kidney tubules. So hopefully you can see with this diagram, water is small enough, so that gets filtered from the blood. Mineral ions like sodium ions are small enough, so they get filtered from the blood. Urea is small enough, so that gets filtered. Glucose is even small enough, so that's getting filtered as well. Amino acids are getting filtered as well. But you can see that these proteins, these large proteins, are too big to pass through the filter, and equally blood cells like your red blood cells or white blood cells are too big to pass through the filter. So they remain in the blood, but these things get filtered via the kidney tubules into the filtrate. Now, we don't want to lose all of these things. We don't want to lose all of the glucose in our urine. We don't want to lose all of our mineral ions in our urine, and we don't want to lose all of our water in our urine. So the kidneys then perform what we call selective reabsorption. So certain things that have been filtered get reabsorbed back into our blood. So all of our glucose should be reabsorbed. It should all go back into the blood. So it does pass through the filter, it does go into the filtrate, but then it will all be reabsorbed back into the blood. The amount of water and ions reabsorbed does vary depending on the body's needs. So a lot of water will be reabsorbed, but excess water will remain in the filtrate. A lot of ions will be reabsorbed, but excess ions will remain in the filtrate. So it's quite a complex process. We say the kidneys filter the blood, but then they reabsorb all of the glucose, some water, and some mineral ions. Now you'll notice no urea is reabsorbed, and that's because urea is a toxic waste product that we need to excrete. So we want all of the urea to be lost in our urine. We don't want to reabsorb it back into our bloodstream. Now, at the end of this complex process, the kidneys produce urine, which then goes to the bladder to be stored before, obviously, we get rid of it or excrete it. So the final urine will contain all urea, excess mineral ions, and excess water. Because remember, a lot of water does get reabsorbed, a lot of mineral ions do get reabsorbed, but there'll be any kind of waste water that we don't need, waste mineral ions that we don't need in our urine. And importantly, all the urea that is filtered from the blood will still be in the urine. It does not get reabsorbed. Okay, so what is urea? Now, this is higher tier only. As I've said already, urea is a toxic waste product, but how is it actually made? Well, when we eat proteins, we digest them into amino acids, which we know from paper 1. Now, if we've got an excess, too many amino acids, the body cannot store excess amino acids. So excess amino acids go to the liver where they are deaminated. So this is a process called deamination, and it occurs in the liver. So excess amino acids are deaminated, which basically means the amino group is removed from the amino acids. Now, you don't have to know that. You can just say excess amino acids are deaminated, which forms ammonia. And then ammonia is highly toxic. So we convert that ammonia into urea. So let me do you a little flow chart, if you like. So excess amino acids that we have from protein digestion, they go to the liver. The liver converts them into ammonia, and then ammonia is converted into urea. So it's the liver that makes urea. Urea is then transported to the kidneys, and as we've just seen, once it gets to the kidneys, it will be filtered from the blood. The kidney tubules will filter it out of the blood. It will go into the filtrate, and then we excrete it in our urine. So we're getting rid of all that toxic urea. Urea is still toxic in large amounts, but less so than ammonia. So when we deaminate the amino acids in the liver, we convert it to ammonia first, then to urea, because urea is less toxic, but still toxic enough that we do need to excrete it in our urine. Now, this again is higher tier. So if you're doing triple foundation, you don't have to know this either. To maintain a constant internal environment, we do have to regulate water content. Now, we spoke earlier about how water is lost from our lungs when we exhale, it's lost from our skin when we sweat. But the only way we can control the amount of water that we lose is via our kidneys using a hormone called ADH. Now, you should know from when you did the endocrine system, ADH, it's a hormone, it's a chemical messenger that travels in the blood. It is produced by the pituitary gland. It does stand for antidiuretic hormone, but you can just call it ADH at GCSE. It's produced by the pituitary gland, which is that P-shaped kind of small part in the middle of your brain, the master gland. And the pituitary gland releases ADH when the blood becomes too concentrated, i.e., there is not enough water in your blood. So maybe you've been exercising, it's been a really hot day, you've been sweating and you've lost a lot of water as it's evaporated in that sweat from your skin, or maybe you've just not been drinking enough water. So your blood becomes really concentrated and your blood does not contain enough water. ADH will then be produced by the pituitary gland. It will travel in the bloodstream to the kidneys and it will act on the kidney tubules. It will make them more permeable to water so that more water gets reabsorbed from the kidney tubules back into the blood. Okay, so sounds complicated, but you can massively simplify this. Okay, so ADH released from the pituitary gland, it makes the kidney tubules more permeable to water, so more water gets reabsorbed. It goes from the kidney tubules back into your blood. Now, if more water is being reabsorbed, this means less water will be lost in your urine. So your urine will be darker in colour, right? It'll be more concentrated because it has less water in it, and you'll produce much smaller volumes of urine. And this is to make sure that you don't lose as much water in your urine because you want to keep more water in your bloodstream. Because remember, in this example, your blood was too concentrated. It didn't have enough water. So we need to reabsorb more water back into the bloodstream. So we lose less water in our urine, producing smaller volumes of more concentrated urine. Let's go through that on a diagram. So if you've got too little water in your blood, or your blood is very concentrated, the hypothalamus will detect, I should have said the hypothalamus contains receptors to detect that there is too little water in your blood, or your blood is too concentrated. That will signal the pituitary gland to release ADH, a hormone, into your bloodstream. That will lead to more water being reabsorbed from your kidneys, because ADH makes kidney tubules more permeable to water. So if the kidney tubules are made more permeable to water, more water will be reabsorbed from the kidneys, which means less water will be lost in your urine, which is why we said urine is more concentrated and there's smaller volumes of it. And that will return your blood water levels back to normal. Now, the opposite side of this, what if you have too much water in your blood? Like, what if your blood is really dilute? You've just drank like 3 litres of water or something and you're not sweating or exercising. Well, again, the receptors in your hypothalamus would detect the water levels. They detect there's too much water in the blood, or the blood is too dilute. The pituitary gland would release less ADH, or stop releasing ADH altogether, which means less water would be reabsorbed from your kidneys, because the kidney tubules would not be made more permeable to water. And if less water gets reabsorbed from the kidney tubules back into the blood, that means more water would be in your urine. So you'd produce larger volumes of urine, and it would be more dilute urine, because you're trying to get rid of more excess water to return your blood water levels back to normal. Okay, let's summarize it, because I know that's really hard. Again, this is higher tier, so it is going to be hard, and it's triple as well. So if your blood is too concentrated, as in there's not enough water in your blood, ADH levels will increase, so your pituitary gland will release more ADH. This means more water will be reabsorbed from the kidney tubules because they're more permeable to water. So less water will be produced, less water will be in your urine. So your urine will have a smaller volume and it will be more concentrated. If your blood is too dilute, so it contains too much water, more water than it should, then ADH secretion decreases. So your pituitary gland releases less ADH, or stops releasing ADH. Less water gets reabsorbed because the kidney tubules are not more permeable to water. So there'll be more water in your urine. So your urine would be more dilute and you'd produce more of it, larger volumes of it. You'd probably urinate more frequently to get rid of that excess water. Now, this is another example of negative feedback, isn't it? We're trying to maintain a stable internal water balance. So we can see that if the water levels in the blood go too high, it's detected, we initiate a response to bring the water levels back down. But equally, if the water levels drop too low in the blood, that's detected and a response is initiated to bring the water levels back up. And this response is all controlled by the pituitary gland and the release of ADH to control how much water gets reabsorbed from your kidney tubules. Right, the next thing, which is for both higher tier and foundation tier triple science, is looking at what happens if kidneys fail. So if someone has kidney disease or kidney failure, we need to look at kidney transplants versus kidney dialysis. And this quite often comes up with like an evaluate style question. So we're going to go through the pros and cons, because kidney failure can lead to death, right? Because if you've got kidney failure, urea, which is toxic, is going to build up in your bloodstream. So it can lead to death unless it is treated with either kidney transplant or kidney dialysis. Now, if we're going to have a kidney transplant, that basically means we're going to replace a failed kidney with a working kidney from a donor. And you can actually get a kidney from a living donor because we do have 2 kidneys and you can survive with just one. So a living donor can donate a kidney. And if this operation is successful, this would be a permanent cure and a permanent solution. Let's go through the advantages of having a kidney transplant. So it's a one-off operation, right? You would not need to go to the hospital and have regular dialysis treatment. So there are far fewer hospital visits involved. If the operation is successful with the donor kidney, you can then lead a more normal lifestyle. You would not have to restrict your diet or restrict your day-to-day activities. In the long term, it is cheaper than kidney dialysis. Even though the operation is expensive, it's a one-off operation. So in the long term, it is cheaper for the NHS or for the hospitals. And the new kidney, if it functions as it should, it will maintain low levels of urea in the blood because it will be filtering the urea out of the blood and will be excreting that urea in our urine. Disadvantages though, there is a risk of rejection. So the immune system may attack the new kidney because it came from a donor. Patients must take immunosuppressant drugs for life. These drugs are designed to suppress or weaken the immune system to stop the immune system from rejecting and destroying the transplanted kidney. But if you weaken your immune system with these drugs, it does put you at risk of other infections. You're not going to be able to fight other infections. There's a limited supply of suitable donors, or there's a long waiting list for a suitable donor. Obviously, it's a major operation, it's a major surgery, so you could talk about surgery risks, risks of infection, risks from the anaesthetic, or potential damage to the new organ as it's being transplanted, and recovery from major surgery can take a long time. So you're going to be using these kind of responses in evaluate style questions. You might just be asked to give the pros and cons of having a kidney transplant, or you might be asked to compare kidney transplants to dialysis. So we'll think about that later too. Kidney dialysis is the second method you need to know. This is a mechanical method of filtering the blood. So it's basically doing the job of the kidneys because the kidneys aren't working properly. So the way this works is the patient's blood is passed through a dialysis machine. Now, a dialysis machine involves basically a load of tubes that the patient's blood flows through, but the walls of the tubes are partially permeable membranes. They're basically mimicking the kidney filters. Outside of the tubes is dialysis fluid, and the dialysis fluid will have no urea in it, but it will have the correct levels of glucose, water, and mineral ions, okay? So just to go through that again, because I know this is complicated, the patient's blood is removed from a blood vessel. It's flowing through these tubes in the dialysis machine, but the tubes are partially permeable. Outside of the tubes in the dialysis fluid, there's no urea, but there will be a lot of urea in the patient's blood. In the dialysis fluid, there's some glucose, some water, and mineral ions, basically the ideal concentrations. Let me show you this on a diagram. So the patient's blood is flowing. This is the dialysis tubing, okay? So it's flowing through the dialysis tubing, and you can see that the dialysis tubing has a partially permeable membrane. Now, because there is no urea in the dialysis fluid, the urea in the patient's blood is gonna diffuse out because there's a high concentration of urea in the patient's blood. There's no urea in the dialysis fluid, so you've got a steep concentration gradient. So as the patient's blood is flowing along the dialysis tubing, the urea will diffuse out of the patient's blood. Obviously, the patient's blood cells are too big to pass through the dialysis membranes, so the red blood cells and white blood cells will remain in the blood, as will large proteins. And then because the dialysis fluid does have some glucose, some water, and some mineral ions, some of the patient's water will move out by osmosis. Some of the patient's mineral ions will move out by diffusion, but only if they're in excess in the patient's blood, okay? Because remember, the dialysis fluid has the ideal concentration of water, the ideal concentration of mineral ions, and the ideal concentration of glucose. So only excess mineral ions will diffuse out, and only excess water will move out by osmosis. There should be no net movement really of glucose because the dialysis fluid has the exact correct concentration of glucose, the same concentration as in the patient's blood. So there should be no net movement of glucose out of the dialysis fluid. I think I've explained that already, but let's go through it and see if the notes make more sense than what I've just said. So urea will diffuse out because you've got high urea in the patient's blood, no urea in the dialysis fluid. Excess ions and excess water will diffuse out. So if there's more in the patient's blood than there is in the dialysis fluid, it will diffuse out through the partially permeable membrane. They're small enough to pass through the membrane and they're going to move down their concentration gradient. Glucose should remain in the blood because there should be the same concentration of glucose in the patient's blood as in the dialysis fluid. So there's no concentration gradient, so no net movement by diffusion. And other useful substances like the red blood cells, the white blood cells, the large proteins, they should remain in the blood too. Proteins and blood cells, obviously, well, they're too big to fit through the partially permeable membranes anyway. So the overall gist of this is everything the patient needs to stay in their blood, like their red blood cells, their proteins, their glucose, all of that should stay in the blood. All of the urea, which is toxic and they want to get rid of, should diffuse out of the blood, and excess water and excess mineral ions should diffuse out of the blood. So these dialysis tubes are basically doing the filtering job of the kidney. This is going to maintain normal blood composition. It's going to remove all the urea from the patient's blood. Now, dialysis is often used when a patient with kidney failure is waiting for a suitable donor for a kidney transplant. Obviously, a kidney transplant is ideal because it is a permanent cure, a permanent solution. But because there is a long waiting list for a suitable donor, a patient might be having dialysis whilst they are on that wait list. Dialysis does keep them alive. It removes toxic urea from their blood. However, it has many disadvantages. So let's go through. So dialysis must be done around 3 times a week. And each session can last for up to 5 hours and is usually done in a hospital. So think about that. That is going to massively affect a patient's quality of life. It's going to put restrictions on activities, restrictions on traveling, going on holiday, or attending work. If patients don't live near to the hospital, then there's a lot of travel time to and from the hospital and a lot of expense for transport to and from the hospital. Now, between the dialysis sessions, urea will start to build up. The concentration of urea will increase in the patient's blood. That can make them feel very unwell, okay? Because they have the dialysis session, which removes all the urea, but as soon as they leave the hospital, urea concentrations start to increase and they might not be going back to the hospital for another 3 days. So levels of urea will gradually build up and could make them start to feel unwell. And this is why you have to have dialysis done very, very regularly as well. Patients need to carefully monitor their diet, so particularly restrict the amount of protein eaten to make sure urea levels do not increase too much. Remember, urea is made from excess amino acids that are deaminated in the liver. So they do need to restrict the amount of protein they eat, otherwise they will end up with more excess amino acids and more urea, and the concentration of urea would increase significantly in their bloodstream. During dialysis, a needle is put into the patient's arm to draw out their blood to move it into the dialysis tubes, so that carries a risk of infection. And you can say patients need to take anti-clotting medication to reduce their risks of developing blood clots because their blood is going to be flowing through really narrow tubing. There is a high risk of blood clots unless they take anti-clotting medication. Now, the way this works is you'll probably have to evaluate either transplants or dialysis or both of them together. So make sure you can give the pros and cons. Obviously, these are all cons of dialysis, but you can use your kind of cons of transplant as advantages of dialysis. Like, this does not require surgery, so there's no major risks from anaesthetic. You don't require a donor kidney, so there's no long wait for a suitable donor. And because you're not having a transplant here, there's no risk of rejection from your immune system. So you can kind of use the cons of transplant as the pros of dialysis, if that makes sense. Or you could use the pros of dialysis, I'm trying to think, as the cons of transplant. So make sure you can kind of give, I don't know, 2 advantages, 2 disadvantages for transplant, 2 advantages, 2 disadvantages for dialysis. Basically make sure you can compare them. But bear in mind, dialysis is usually done whilst waiting for a suitable donor kidney for transplant, because this is not a long-term solution. This is not a cure. So that's a big pro of a kidney transplant. That is a long-term solution. That is a permanent cure. And that would mean you would have no restrictions on activities and your kidney would be working all the time. So levels of urea would not build up in between sessions like they do with dialysis. Okay, so now we are on to our plant hormones, and we're going to be starting with tropisms. So what are tropisms? Tropisms are directional growth responses of a plant to a stimulus. They're directional because the direction of growth is determined by the direction of the stimulus. So let's look at the types. We have phototropism, which is the directional growth response to light as a stimulus. And we have gravitropism, also known as geotropism, you can call it either, which is the directional growth response to gravity. Now there's other types as well, like hydrotropism is the directional growth response to water. But these are the 2 that you need to know for GCSE. Now, something else you need to appreciate is we can have positive and negative tropisms. So a positive tropism is where the directional growth is towards the stimulus, and a negative tropism is where the directional growth is away from the stimulus. So for example, putting all of this together, the shoots show positive phototropism because the shoots grow towards the stimulus of light, whereas the roots show negative phototropism because the roots grow away from the stimulus of light. They grow down into the soil, don't they? If we think about gravity, the shoots show negative gravitropism because they grow away from the force of gravity because they grow up, whereas the roots show positive gravitropism because they grow down towards the stimulus of gravity. Now, how is this all controlled and coordinated? Well, it's controlled by a hormone called auxin. So auxin is a plant hormone and it controls growth near the tips of the roots and the shoots, because auxin is produced in shoot tips and root tips. So it's produced, here we've got a shoot, right? It's produced in the tip of the shoot and it's also produced in the tip of the root. When it's produced, it will then move by diffusion. So it will start to diffuse from a higher concentration of auxin to a lower concentration of auxin. And when it diffuses, you can get uneven distribution of auxin, and that will cause unequal growth rates. So let's have a look at a couple of different scenarios. So I want you to imagine you've got a shoot growing in even light, so equal light from all around. The auxin, the plant hormone, which is produced in the shoot tip, will start to diffuse from high to low concentration, and it will spread out evenly in the shoot because the light is equal. So the auxin will diffuse, you'll get even distribution of auxin, which means you get equal growth. So the shoot will grow evenly on both sides and the shoot will just grow straight up, okay? But let's have a look at another scenario. So let's imagine now we've got light from one direction, coming from the side only. Now again, we'd say auxin is produced in the shoot tip. Auxin will start to diffuse down the shoot from a higher concentration in the tip to a lower concentration down the shoot. But because we've got light coming from here, the auxin will diffuse and it will move towards the shaded side. I always think of it like auxin likes the dark side. It goes to the dark side. Is that Star Wars? I think that's Star Wars. Goes to the dark side. So auxin diffuses and it accumulates or builds up on the shaded side. There's a higher concentration of auxin on the shaded side. And this means that the shaded side grows faster than the brightly lit side because the cells will get longer on the shaded side. So if you imagine this side is growing faster than this side, that causes the shoot to bend towards the light. I'm going to show you this with my hands. This is what I do in lessons. So if you've got more auxin on this side and auxin is stimulating growth in the shoots, this is the shaded side. So auxin accumulates on the shaded side, or you've got a higher concentration of auxin on the shaded side. So this side grows faster, which bends the shoot towards the light, like that. That is positive phototropism because it's bending and growing towards the stimulus of light. Now, obviously, this is an advantage to a plant that's growing in light from one direction because it will bend towards the light or grow towards the light, and that will increase the amount of light that it's able to absorb for photosynthesis. Okay, so make sure you know here it's auxin that controls tropisms. Tropisms are directional growth responses to a stimulus. Auxin is produced in shoot tips. It stimulates growth in shoots. If you have equal light, the auxin will distribute evenly, so you'll get equal growth. The shoot will grow straight up. But if you've got light from one direction, the auxin accumulates on the shaded side, so you've got an unequal distribution of auxin. So the shaded side grows faster, which is why the shoot bends towards the light. Now we need to think about gravitropism as well. Gravitropism is the growth response to the force of gravity. So gravity is the stimulus. Now, if you think about in a root, let me do you a little picture. Let's draw a little seed. And when a seed starts to germinate or sprout, the roots will start to grow out of one side. Now, because of the force of gravity, let me make that a bit bigger. Here's my seed. Here's my root that starts to grow. Because of the force of gravity, auxin will accumulate on the lower side of the root. So again, auxin is produced in the tip of the root, just like it's produced in the tip of the shoot. Auxin will start to diffuse down the root from a higher concentration to a lower concentration. But because of the force of gravity, auxin will accumulate on the lower side of the root. So again, you've got an uneven distribution of auxin. And you can see it on this big diagram here. So you can see auxin is produced in the root tip. It's going to start to diffuse down the root, but you'll get auxin accumulating on the lower side of the root due to gravity. You can see there's a higher concentration of auxin on the lower side of the root compared to the upper side of the root. Now, what's a bit weird that we must remember about this topic, and I'll write it nice and big, in roots, high concentrations of auxin, they don't stimulate growth, they inhibit growth. So the effects of auxin are opposite in shoots compared to roots. So in shoots, where you have a high concentration of auxin, like on the shaded side of a shoot, it stimulates growth. But in roots, where you have a high concentration of auxin, which would be the lower side of the root, it inhibits growth. Okay? So what's going to actually happen here is the upper surface of the root will grow faster because the lower surface of the root growth is being inhibited or restricted by auxin. So the upper surface will grow faster. And again, I'll show you with my hands. If the upper surface grows faster, it's going to cause the root to bend down towards the stimulus, which is gravity. Okay, so the lower side has a higher concentration of auxin. High concentration of auxin in roots actually inhibits growth. So the upper surface, where there's less auxin, is growing faster because the cells are elongating faster. The upper surface grows faster and it pulls the root down towards the stimulus, towards the force of gravity. Now, again, this benefits the plant because we want the roots to grow down into the soil for a few reasons. It will help to anchor the plant in the soil. And obviously, roots are where plants absorb water by osmosis and mineral ions, such as nitrate ions, by active transport. So we want them to grow down. Now, I'm just going to add here, because I think it's super important. In roots, high concentrations of auxin inhibit growth, but in shoots, it's the opposite. So where you see the high concentration of auxin, it stimulates growth. So it has the opposite effect in roots and shoots. And obviously shoots show positive phototropism, whereas roots show positive gravitropism. So the roots will grow down towards gravity, the shoots will grow up towards light. Now there is a practical that has come up a lot for triple science. The aim of the practical, the required practical, is to investigate the effect of light or gravity on some germinating seedlings. Now, I'm going to give you an example method, and in my method, I'm investigating the effect of light on germinated seedlings because this has come up like twice in recent years for triple science. So what you could do, get your Petri dish and line it with moist cotton wool, because obviously they do need some water to start to germinate. Take 10 seeds and evenly space them on the surface of the cotton wool. Cover the dish with a box that has a hole cut in one side. This is to make sure that light comes in from one direction only, so you can investigate the effect of light from one direction. So you could even shine a lamp into the hole in the box, so light is coming in just from one direction. Place it in a warm place, so warmish temperature, so the seeds start to germinate. Leave them for a set amount of time, so I've suggested 5 days. You could then measure the length of the shoots and observe the direction of growth or the bending of the shoots. When you're measuring the shoots, it is really hard to do because they are bending towards the light. So you could suggest use forceps to carefully straighten the shoots to help you measure them accurately with a ruler. Or the other thing you could do is use a piece of string placed next to the shoots. So kind of measure it with the string, because string is bendy, and then place your string on the ruler to measure the length of the shoots. I mean, you can just say use your fingers to straighten the shoots so you can measure them accurately. Or maybe even use one of those flexible tape measures that bends so you can measure them accurately. You could also draw some biological diagrams of the seedlings to actually show the curvature. That's optional. You might have done that in school, you might not. And then what you'd have to do, because so far you've only done it in one light condition, you've only done it with light from one direction. So you'd have to repeat this with other conditions, such as no cardboard box, making sure that light is even, so you can compare the growth and the curvature of your seedlings that had light from one direction with your seedlings that were in even light. You could even compare them with seedlings that you'd placed in a cardboard box with no hole, so it was completely dark. So you've got 3 different light conditions: light from one direction, no light, and even light. Now, in this experiment, we started with seeds that hadn't yet germinated. But in your exam, you might be provided with a picture that shows you seedlings that have already started to grow. If the seedlings have already started to grow, then what you could suggest doing is measure the length of the seedlings before you start the investigation, and then measure the length of the seedlings after the investigation, because then you could calculate the increase in length, the mean increase as well. Because remember, you would not just have one seedling in each condition, you want as many seedlings as possible so that you've got multiple repeats. So we had 10 seeds on the surface of the cotton wool in the Petri dish. You could do 10 seeds in light from one direction, 10 seeds in total darkness, 10 seeds in even light, measure the length of the seedlings before and after, work out the mean, the average increase in length in each of those three conditions. Now, as always, when we go through practicals, and I do it all the time in these videos, your examiner is going to be kind of reading your answer if you're planning a practical, and you really want to highlight what is your independent variable, so what are you changing? What is your dependent variable, so what are you measuring or observing? And what do you need to control so that you get valid results? So the independent variable in this required practical, because we're investigating the effect of light intensity, the independent variable, the thing we're changing, is the different light conditions. So you have to make it clear in your answer that you are looking at different light conditions. So one directional light, which is your cardboard box with a hole with a lamp shining. Even light, so plants just out in the open with light from all around. And then you could even maybe do complete darkness as well. So you've got three different versions. That's what you're changing. That's your independent variable. Dependent variable, you're going to say what you're going to measure. So you could say, I'm going to measure the lengths of the shoot using a ruler. And you're probably going to have to do this before starting the experiment and after the experiment's finished so that you can calculate the increase in length. But for this, you could also say, record the direction of growth. So you could write down how much the shoots are bending by. You could take photos of the shoots to show the curvature. You could do drawings of the shoots. I mean, you could suggest trying to measure the angle of bending with a protractor. It would be very tricky to do, but you could suggest that as one of your dependent variables. Control variables, remember, we need to give at least 3. So if you've got 3 Petri dishes of seedlings in 3 different light conditions, what would you have to keep the same so that it did not affect their growth and you got valid results? Well, same volume of water on the cotton wool, same amount or same volume of water, same type of seedling, so all cress seedlings or all grass seedlings, same age of seedlings at the start. So are you going to use a seedling that has already started to germinate and grow, or are you going to use a seed that has not yet started to germinate? You need to start them off at the same point. Same temperature, because temperature would affect the growth of the seedlings. And really importantly, the same time that the seedlings are left to grow. So 5 days, I think we had in our method. You can't leave one in a box for 10 days and the other one in open light for 4 days, because obviously the length of time will affect the amount of growth. So always try to give at least 3 control variables. And what I do when I'm writing a method is I make it very clear. I say, this is what I'm changing. So I'll have one in a box that's dark, one in open light, and one in a box with a hole cut in the side in one directional light. Make it very, very clear. Then I say, this is what I'm measuring, and this is how I'm going to do it. And this is what I'm controlling to make sure I get valid results. The other thing actually that's not on there is repeats, because we say IV, DV, CV, repeats. IV, DV, CV, repeats. Remember that, I've said it so many times. But don't just do this with one seedling. I think we had 10 seedlings in each condition, because then what that enables you to do is to get reliable data, because you could measure the before height and the after height of all 10 seedlings in each condition, and then you could work out the mean increase in length. Always better. The more repeats, the better. So 10 seedlings in each condition would be a really good suggestion for this practical. Okay, final thing, and this is higher tier only in Module 5. We've spoken about auxin as a plant hormone, but there are other plant hormones, and we need to look at how they are used in agriculture or farming and horticulture or gardening. So we're going to look at commercial uses of auxins, but we're also going to look at two other plant hormones, gibberellins and ethene. Now, obviously, we've already looked at auxins. So quick recap, we know that auxins are produced in the tips of the roots and the tips of the shoots. We know that auxins control tropisms, like phototropism, growth towards light, gravitropism, growth towards gravity. And we also know that where there's a higher concentration of auxin in shoots, it stimulates growth, and where there's a higher concentration of auxin in roots, it inhibits growth. So you learn about your auxins in relation to your tropisms, but these are commercial uses of auxins. So where we use auxins in other areas of agriculture and horticulture. So we can use auxins in weed killers. Why? You might think auxins are good, right? They stimulate growth in shoots, but some weed killers contain really, really high concentrations of auxin. Basically, that causes the shoots to grow so quickly in an uncontrolled manner, like rapid cell elongation. They grow so quickly that it kills the plant. So you can put really high concentrations of auxin into your weed killers. We also put auxins into rooting powders. Now, rooting powders are used when you take plant cuttings, which is a type of plant cloning, another triple science topic from Module 6. But if you take a cutting of a side shoot, say, from a plant, you can dip the cut end into auxins to encourage root growth. So we say we use auxins in rooting powders when cloning plants or taking cuttings to encourage the growth of roots. Another type of plant cloning, which you do in Module 6 if you're triple science, is tissue culture. Now, tissue culture is where you just take a few cells from a plant, say from a leaf, and you can grow a whole new plant just from a small number of cells. But when we're doing that, we add auxins to their growth medium to stimulate growth of the cells into a full plant. Okay, so 3 commercial uses of auxins. Obviously, they control tropisms, but we can also use them in weed killers because they cause such rapid growth that the plant dies. And we can use them in both of these methods for cloning plants. We add them to rooting powder when we take cuttings, and we add them to the growth medium when we carry out tissue culture. Now, the next hormone we're going to talk about is gibberellins. Again, these can be used commercially in agriculture and horticulture. Now, what do gibberellins do, and why would we add gibberellins to the growth medium of a plant? So the first thing they do is they end seed dormancy. Basically, adding gibberellins helps seeds to germinate or start to grow, start to sprout. And if you add gibberellins, you can get seeds to germinate earlier than they normally would. So normally they start to germinate when the temperature increases, right, in spring, mid-spring. But if you add gibberellins, you can get them to germinate even when the temperatures are cold. So they can germinate earlier in the year, like early at the start of spring. So their growing season is longer, so they grow bigger. So that's one thing you can say about gibberellins. They stimulate germination of a seed. They also promote flowering. So they speed up flowering or trigger flowering. And again, you can also say that leads to larger flowers, right? If you stimulate flowering, and you can stimulate flowering again at any time, regardless of the temperature or light conditions, you can use gibberellins to promote flowering. And the third one is using gibberellins to increase the size of fruits, make the fruits grow bigger, so we get larger, more marketable fruit that consumers want to buy. This is particularly useful for seedless fruits, so like Seedless varieties of grapes do tend to grow quite small. So if you add gibberellins, the seedless grapes will grow larger, which might be more appealing to consumers that buy them in supermarkets. The last plant hormone, and again, this is higher tier only, is ethene. And the commercial use of ethene that we need to know is ethene controls fruit ripening. So ethene gas basically promotes ripening. It starts off the ripening process. Ethene gas is used in transport to ripen fruit just before sale. So just before the fruit goes onto the supermarket shelves, it will be put into an environment with ethene gas to cause it to ripen so that, I don't know, bananas are yellow when they go onto the shelves. It says here it prevents spoilage during shipping. Let me explain that. What we want to do is we want to transport the fruit when it's unripe. Because think about an unripe banana. It's really green, it's firm, it won't be damaged during transport because it's much harder. So we transport them unripe, but then we add the ethene gas to ripen them quickly just before we put them onto the shelves for consumers to buy. So I've given you an example here. Bananas are picked and transported green, unripe, to prevent damage, and then we ripen them with ethene on arrival at the supermarket so that consumers actually want to buy them. And the other thing that I should just add here, because it's not on the slide, as well as ethene controlling ripening of fruit, we can also say for ethene, it does help to control cell division. So those are the two things on your spec that it says about ethene. Controls cell division, controls ripening of fruit. And then we had ending seed dormancy, promoting flowering or getting larger flowers for gibberellins, larger fruit for gibberellins. And then we had weed killers, rooting powder, and used in tissue culture for auxins. So make sure you basically know, if you're triple science and higher tier, you know the names of these three hormones and you can give these commercial uses of the hormones. And that is it.
Thank you for watching, and as always, I hope you found that really useful for your revision. Make sure you click the link in the description to access those free flashcards on these triple science only topics. And also there's a link in the description if you want to access my resources where you can find everything, study notes, videos, practice questions, everything you need to revise GCSE biology with us here at MyEdSpace. Make sure you're subscribed because we're going to be posting more videos just like this very, very soon.